Hey guys, welcome back to week 3. This week we're going to talk about nucleic acids and DNA. Before we go into that, let me ask you a question. Have you ever sat in an examination, flipped open your page, the extended response, and actually find yourself writing lines and lines and lines of stuff and then ending up getting no points? Today I'm going to teach you how to use a table format to accurately and nicely present your answers so you're on point and you know what you've written. First up, nucleic acids, just like proteins, are also macromolecules. They store genetic information and are involved in the production of proteins. For example, we have DNA and RNA. Just briefly introducing the structure of nucleic acids, DNA and RNA, the nucleic acids, are actually made up of smaller subunits or monomers. We call these monomers nucleotides. In the right-hand corner, you can see a nucleotide. So each nucleotide is actually composed of a collection of things. In the center, you can see a 5-carbon sugar. So the 5-carbon sugar has 2 arms. It likes to hang on to 2 friends. On the left, in this picture, you can see a phosphate group. And on the right, in this picture, you can see a nitrogenous base. There are 4 different types of nitrogenous bases that a 5-carbon sugar can possibly link to for DNA. They include adenine, cytosine, guanine, and thymine. Alright, so on the left you can see a strand of DNA, or it can also be a strand of RNA. In this particular example, we have 4 nucleotides forming part of a strand, and we call this a macromolecule. Today, the question we're going to discuss is contrast the structural characteristics of RNA with DNA. And I'm going to guide you through this question. I have circled and underlined the key points of this question, and note that this question is a 3-mark question, meaning you should give at least 3 differences between RNA and DNA. Today, we're going to draw a table. On the left-hand side of this table, in the left column, we have all the characteristics we're going to compare between DNA and RNA, and they are nitrogenous bases, type of 5-carbon sugar, and number of strands. So first up, let's fill in the boxes one by one. For DNA, we have adenine, guanine, thymine, and cytosine, as I've mentioned before. For RNA, it's basically the same, except that the base thymine is actually replaced by uracil, meaning that RNA has 4 types of bases, and they are adenine, guanine, uracil, and cytosine. As for the type of 5-carbon sugar that DNA and RNA have, DNA is actually named deoxyribonucleic acid in its full name, and therefore its 5-carbon sugar is deoxyribose. For RNA, RNA's full name is actually ribonucleic acids, therefore its 5-carbon sugar is ribose. For DNA, the number of strands is actually 2. DNA is arranged in a double helix format, and therefore there are 2 strands of DNA that are paired together to make one entire DNA molecule. RNA is actually single-stranded. So say if I were to present my answer in a paragraph format, this is how I would do it. I would encourage you to pause this video and actually have a look at the answer. But I can tell you that in this answer, this student has repeated the terms RNA and DNA both 5 times. Say if this question was actually contrast the structural characteristics of proteins with carbohydrates, you would have to repeat these very lengthy terms for a lot of times, which is not very ideal when you're in time-constrained conditions. This is why I would like you to actually practice using tables to answer these questions, to make it succinct and very nicely presented. The question for today is to compare the structural and functional features of mRNA, rRNA, and tRNA. I would like you to present your answers in a table. Note that you need to do some research on what different types of RNA molecules do in order to answer this question, and we'll discuss in class.